this is Dr. Bond, and we're going to talk about the electron transport chain and chemiosmosis in this series.
I don't like the citric acid cycle. It seems very difficult.
Okay, all three of these mean the same thing. We have Krebs cycle because the Krebs cycle was discovered by Adolf Kreb. We have the TCA, tricarboxylic acid cycle, because there are 3 carboxylate groups on the intermediates. And then we have the first intermediate known as citric acid. So we can also call it the CAC, or the citric acid cycle. And before we get into the citric acid cycle, we just went over pyruvate to acetyl-CoA. We have lost CO2. We have reduced a NAD, and this is inhibited by ATP, acetyl-CoA. So we have end product inhibition and the reduced form of NAD. Okay, so this reaction is inhibited by ATP, which is a sign of too much energy. So is NADH, too much energy, and end product inhibition with acetyl-CoA. So acetyl-CoA will enter the cycle, and what we're going to bond with is a 4-carbon oxaloacetate. We're going to form citric acid, and acetyl-CoA will go away. We lose acetyl-CoA. The enzyme is citrate synthase, and a synthase gets its energy from another source without having to break a phosphate, an anhydride bond. It doesn't use ATP to build something. So this is going to actually be using the thioester bond from this guy. When we break off the coenzyme A from the acetyl group, we're going to use that energy, and this is highly exergonic, K at negative 32.8 kilojoules per mole. So this is actually going to be driving pulling the cycle. This reaction is inhibited by ATP and NADH, which is low energy, as well as citric acid, or citrate. Let's just go ahead and change that to citrate. And Succinyl-CoA, which is going to be a step down here we'll talk about. Okay, so we're going to be inhibited. This is an allosteric enzyme that will be inhibited by ATP, NADH, citrate, and succinyl-CoA. Citrate being the end product inhibition, and succinyl-CoA just being an allosteric inhibitor. Okay, so we have a 6-carbon molecule here, and remember this was 2-carbon. The next step, this is an isomerization. It is catalyzed by a conatase. We are creating a chiral compound known as isocitrate, which is still 6-carbon. There is a quick intermediate in here. It's called cis-aconitase, or aconitase. This is where we get the enzyme name from. Okay, so this is step 1. This is step 2. Step 3, we're going from isocitrate to alpha ketoglutarate, which is a 5-carbon molecule. So we're going to be losing a CO2. This is an oxidative decarboxylation. That is isocitrate dehydrogenase enzyme that catalyzes this reaction. We do get a reduced NAD because this is an oxidative step. So it's oxidative and there's decarboxylation occurring. So we go from a 6-carbon to a 5-carbon alpha ketoglutarate. The next step, we are going from a 5-carbon to a 4-carbon. This is Succinate-CoA, which was an allosteric inhibitor of this first step, of step number 1. Here we have alpha ketoglutarate dehydrogenase complex. This is the enzyme that was like the pyruvate dehydrogenase complex. So it has multiple coenzymes. We have TPP, lipoic acid, we have magnesium, we have BAD, and then we have coenzyme A. From here, so now, we're at four carbons, so the rest of the cycle is getting back to oxaloacetate, but on the way we're going to reduce carriers and create ATP, which happens in the next step. Succinyl-CoA to succinate, we're going to lose a coenzyme A. We're going to create a GTP, which we can convert with another enzyme very easily to ATP. So that's why we can say for each turn of the cycle we're getting an ATP. This enzyme is called Succinyl-CoA synthetase, and this one is named, the rest of them are kind of intuitive except for aconitase. This is named for the intermediate step, but this one is named for the reverse reaction because it was discovered first. But a synthetase enzyme, unlike a synthase enzyme, this is going to require breaking a phosphate bond and an anhydride bond, like of ATP, or in this case it would be of GTP. So if you look at the reverse reaction, we're going from GTP to GDP over here. So that's why this is named a synthetase enzyme, not because of this reaction, but because of the reverse reaction. So here we're left with a succinate, which is 4 carbons. We've lost a coenzyme A. Again, we can use the energy in breaking this bond between the succinate and CoA, because it's a thioester bond that has a lot of energy. And ultimately, this is exergonic, even though we're creating ATP. It's just slightly exergonic when you do the math, but exergonic nonetheless. So the next step, this step has a claim to fame of being the only enzyme that's in the inner mitochondrial membrane. It is called Succinate Dehydrogenase. We're going to tweak this term when we talk about the electron transport system, but for the citric acid cycle, this is enough. We call it Succinate Dehydrogenase. Just know there's a little bit more to it. We're still going to a 4-carbon. This one is an oxidation step. This is a fad-linked oxidation. And I should go back and say this right here, this is substrate level phosphorylation, if you remember that term from glycolysis. And right here, oh, I forgot to add this too. This is a NAD, so this is an oxidative. Oh, I said it here. Oh, no, this one is also. Okay, so step 3 is an oxidative decarboxylation. Step 4 is an oxidative decarboxylation. Step 5 is a substrate level phosphorylation. Step 6 is a fad-linked oxidation. There's no more decarboxylation. The decarboxylation steps were here and here, steps 3 and 4. All right, from here we have a fumarate molecule. We just need to get it to oxaloacetate. So we're going to add water. So this is a hydration step, and this is going to give us malate, L-malate to be specific. This is done by fumarase. So this is a hydration. And the next step is going to be another oxidation. Okay, so this is an oxidation step from L-malate to oxaloacetate, and this is going to be catalyzed by malate dehydrogenase. In summary, for the citric acid cycle, we have 8 steps, 8 steps, and we enter with acetyl-CoA, which is a 2-carbon molecule. It's going to combine with a 4-carbon oxaloacetate. This step is inhibited by ATP, NADH, citrate, and succinyl-CoA. We are going to create citrate as our first step. So citrate is our first intermediate. It's 6-carbon. We lose our coenzyme A of this reaction. What we're using for the energy to drive this synthase reaction is breaking the thioester bond of this coenzyme A. Coenzyme A is an activator. When it's bound to something, it is known as an activation step. So this acetyl group is activated. So when we break this thioester bond, we have a large exergonic reaction. We're creating a citrate. Step 2, we're creating a chiral. Citrate is achiral. This is chiral. There's actually 4 different isomers of it, but only one's going to be produced, which is this isocitrate. This reaction is catalyzed by a aconitase because there's a quick intermediate step where we have a cis-aconitase created, but the ultimate product is the isocitrate. From here, isocitrate dehydrogenase, this is a oxidative decarboxylation step. This is going to be giving off a CO2 and reducing a NAD, and this is an electron carrier that will go to the ETS. Step number 3 is under regulatory control, and step 3 is going to be inhibited by ATP and NADH and activated by ADP and NAD, the oxidized form. Okay, so that's step 3. There's a regulatory step. Okay, so step 3 is regulated. Step 4 is also regulated. Step 4 is going to be inhibited by ATP, NADH, and succinyl-CoA. So we have some end product, because this is the end product inhibition for step 4. So steps 1, 3, and 4 are the regulatory steps of the citric acid cycle. Alright, step 4 is going to be catalyzed by this huge complex known as alpha-ketoglutarate dehydrogenase complex. Step 4 goes from alpha-ketoglutarate to succinyl-CoA. We are losing a CO2, so we go from a 5-carbon to a 4-carbon. The succinyl-CoA is going to be, therefore, in step 5, substrate level phosphorylation. We produce GTP. This is catalyzed by succinyl-CoA enzyme A synthetase. From here, succinate to fumarate is our next step. This one was the only one in the inner mitochondrial membrane. This one has a FAD-linked oxidation, so we're going to be left with FADH2. From here, we have fumarate, which is 4-carbon still. We're going to be catalyzed by fumarase. It's a hydration step to L-malate. L-malate, again, which is 4-carbon. We're going to go from L-malate to our starting point, which is oxaloacetate with malate dehydrogenase, with an oxidation step. So that's our citric acid cycle.
Okay, so there's 8 steps of the Krebs cycle. I get it.
The electron transport system, the whole function of this is for the electron carriers that have been reduced, the NADH and FADH2. These are going to be dropping off to the ETS. So let's look at what this is. If this is our mitochondria, what we're looking at is this piece right here. Okay, so let's look at this piece. Let's blow it up. Here we have our 2 membranes. Here, I'll even draw this one with the cristae a little bit. So this is inner, outer, and okay, so we have a series of what are known as redox reactions where we have NADH dropping off the electrons. The electrons are going to be dropped off in pairs. It will drop off 2 electrons to complex 1, and I'll discuss the details of that in just a second. And by doing so, we'll be pumping out hydrogens into the inner membrane space against their concentration gradient. Now, FADH2 will drop off the 2 electrons into complex 2 of the ETS, but this one is not a proton pump. What we do have is a coenzyme Q, which is a mobile carrier. This guy is going to be accepting the electrons from both 1 and 2, and it will be, okay, so this coenzyme Q will be dropping off electrons to complex 3, which is another proton pump, and we have another one, cytochrome C, which is another mobile electron carrier, and it will be dropping off at complex 4, and we have proton pumps here. So we have 3 proton pumps. We have complex 1, complex 3, and complex 4. The reason why FAD does not produce as much ATP is because we're dropping off here instead of at proton pump number 1, so it skips that because this is embedded in the inner mitochondrial membrane, and so it just directly drops off to complex 2. Both of these give to coenzyme Q, which thereby gives it to 3. FAD is only contributing to the electrochemical gradient through pumps 3 and 4. Okay, so let's blow this up and see exactly what's going on here. Okay, we have NAD dropping off at complex 1. The first complex is called NADH coenzyme Q reductase. So I'm going to draw it like this. For E is for enzyme. We have an FMN group as a coenzyme in this reaction. And this reaction we're picking up 2 electrons, we're becoming reduced, and this is the oxidized. So in the first step of this reaction, we're going to become reduced. Then we have an iron-sulfur complex, and the electrons will be quickly transferred to the iron-sulfur complex. The thing about the iron-sulfur complex is that it does not want hydrogen. So what it does is it becomes a pump, and it's going to pump into the intermembrane space. So FMN will accept the hydrogens, but when we get here to the iron-sulfur cluster, we're done. So we're going to say hydrogens are gone. Now, hydrogen ions, I should be specific. From here, we have coenzyme Q. This is actually the next step, but coenzyme Q is kind of like a go-between complex 1 and 3. So let's just finish this story. Coenzyme Q is going to pick up 2 electrons, but it is also going to pick up 2 hydrogen ions from the matrix. Okay. And we'll end with that story. This is how NAD, the reduced form of NAD, drops off the electrons. First picked up by the FMN group, which is a derivative of riboflavin, which is closely related to FAD, actually. Here we have a quickly oxidized FMN group. When it gives the electrons to the iron-sulfur complex, this will not accept the hydrogen, so the hydrogens get pumped into the intermembrane space, and we have 2 electrons to give to coenzyme Q. Coenzyme Q is going to be reduced, and this is the reduced form, and this is the oxidized. Complex 2 is not a pump, but what we have going on, if you remember from succinate to fumarate, this was from the citric acid cycle, and we're going to reduce FAD. This happened in the citric acid cycle. From here, this is a cycle, oxidized iron-sulfur cluster. So we're going to pass these electrons that FAD picked up. All of this, remember, this was the one step that had no mobility. The electron carrier is in the inner mitochondrial membrane. It never left because it's just going to transfer its 2 electrons to the iron-sulfur complex of this enzyme. This enzyme we're going to rename to... This enzyme now, we're going to refer to it when we're talking about the ETS, because we're adding this succinate coenzyme Q oxidoreductase. This is complex 2. It is not an electron pump. It doesn't have enough energy to do so. So this is all it is. So it's still going to be giving 2 electrons to coenzyme Q, just like the complex 1 did from NAD. Okay, coenzyme Q is also known as ubiquinone. This is a mobile electron carrier as far as in the inner membrane. Okay, so here we have, okay, we know that we end up with this from complex 1 or 2. The tricky thing from this point, this guy is going to go to complex 3. Okay, so this goes to complex 3. This is pretty much all that this does. So it's going to accept electrons from complex 1 and 2, and it is going to travel in the inner membrane to complex 3. So let's draw out what happens in complex 3. We are going to transfer 1 electron to an iron sulfur complex. We are then going to transfer 1 electron only at a time, to cytochrome C1 oxidase, or cytochrome C1. Cytochrome C1 is going to give it to another mobile carrier. Okay, this is a mobile. So let's go over this. Here we have a coenzyme QH2. It is going to drop off. 1 electron is going to go this route. Another electron is going to go to the Q cycle. We'll discuss this in a minute. Let's just pretend that doesn't happen. So let's say 1 electron is going this route. So we're going to reduce an iron sulfur cluster, which is thereby going to reduce a cytochrome C1. Cytochrome C1 can only take 1 electron at a time, so it has to work double hard. And because it can only take 1 at a time, the other electron is going to go into this Q cycle that we'll discuss. So here we have 1 electron at a time. We're going to be transferring it to a mobile electron carrier in the inner mitochondrial membrane, the second one, the other one being coenzyme Q. And we're going to reduce the cytochrome C, which is going to carry it to complex 4. Now let's talk about this Q cycle.